Recording in progress.
Good morning. I am Namrata A Gaikwad from Dr. Patangrao Kadam Mahavidyalaya Ramanand Nagar Burli. I am here to present my poster on title Impact of Iron Oxide Alpha Fe2O3 Nanoparticles on Garlic Crop Productivity. Let me share the screen. So, as discussed, the title of my topic is Impact of Iron Oxide Alpha Fe2O3 Nanoparticles on Garlic Crop Productivity. We know that nanoparticles have been playing a huge role in many ways in our lives. It has it has a large surface area. It has good chemical reactivity, and its industrial applications have been checked. And recently, or in recent days, we can see that people or the researchers are trying to study the dose response relationships, the soil plant interactions, and environmental safety of various nanoparticles, of which I am studying that for iron oxide. The objectives basically of my study are to synthesize iron oxide nanoparticles by chemical route in controlled laboratory protocol. I have characterized those synthesized nanoparticles, and then I have evaluated the impact of Fe2O3 nanoparticles on the morphological parameters of garlic, which is Alivum sativum, named by Linus. The local variant that I have used is Jumbo Shera, which is locally available in the markets of Sangli district. The methodology that I have followed for performing this experiment is synthesis of nanoparticles, checking its morphology, checking its purity, its elemental composition with the help of X-ray diffraction, with the help of um, FTIR, EDAX, and FESEM. And then we have applied these nanoparticles onto garlic seeds, garlic cloves, let's say. And those garlic cloves, I have initially had seed priming of those garlic cloves into various concentrations of Fe2O3, and further, as the progress of the plant continued for next few weeks, I performed foliar application of the same nanoparticles. So this is the setup of synthesis that I have performed. Basically, I went with ferrous chloride anhydrous, which was dissolved in double distilled water, performing continuous stirring. And then we added NaOH dropwise, which resulted into formation of dark brown precipitation. This dark brown precipitation was further filtrated with the help of Whatman filter paper number 41. This filtrate was further dried. It was dried at 60 degrees Celsius overnight, and then annealing was performed for 1 hour at 400 degrees Celsius, which resulted into formation of iron oxide nanoparticles. This iron oxide nanoparticles, I have uh, changed the variation. I have changed the molarity, and I have 5 replicates of Fe2O3. Since I have changed the concentration, I got different particle sizes. The particle sizes and the characterizations that I performed were XRD, EDAX, and FESEM. XRD itself shows that the JCPDS number 000033306664 matches exactly the synthesis of Fe2O3. As we can see, the peaks show that the nanoparticles that were formulated are in spear shaped. Then to check the chemical composition, we went on to perform EDAX, which showed that almost 70% of atomic weight is of oxygen, whereas 29.87% weight is of Fe, which clearly signifies that Fe2O3 that we have synthesized is rich in oxygen. Further, to check the morphology of the synthesized nanoparticles, I performed FESEM, which showed that the nanoparticles synthesized were well in range of 50 to 110 nanometers, which is acceptable by literature point of view as nanoparticles. So this is the characterization that I underwent. So this is the experimental setup that I'm talking about. We had different particle sizes of Fe2O3. Each particle size was further distributed into different concentrations. This Fe2O3 nanoparticles were dissolved in distilled water. So we had 5 concentrations that we used for plant treatment. One was 0 mg per liter, 25 mg per liter, 50 mg per liter, 100 mg per liter, and 500 mg per liter. And basically, when you look at these concentrations, you'll get to know that each concentration, the 0 mg per liter concentration, is for control, that we need to check our results in comparison of other concentrations. So for each concentration, I have used 5 plants, so 5 plants per treatment. And this way, I have performed experiment on about one particle size, which is further distributed into 5 ion concentrations, and which is further which has further treated 5 plants. So around 300 plants were treated in whole of the experiment. So these are the results of field experiment that I have performed. Initially, as I said, I had used seed priming, where I had where I had poured Fe2O3 solutions of different concentrations into the seeds that were kept for germination. Once germination started, I shifted those saplings to these boils that were collected from a nursery. Then these saplings were further transferred to the bags, which have a diameter of around 25 centimeters. A 5 plant set in each pot, in each black bag, were planted. And this was the growth that I observed in the first month after sowing. The results are further presented in the graphs. And at the harvest, this was the morphology of the plants that were harvested in the month of April last year. This is the evaluation data, where we can see I have plotted a graph of number of leaves versus concentration. What you can see here is for first 3 sets, the data shows enhanced leaf production, indicating chlorophyll biosynthesis, photosynthetic efficiency, and metabolic activity. Further, for set 4 and 5, we can see that this performance has reduced. The red marked pointer shows the control, which is 0 mg per liter. The same experiment was performed, the same data analysis was done for plant height versus concentration. What you can see here is this graph is plotted as scatter plot. One way ANOVA has been performed and Turkey HSD test has been performed. And this graph, it is plotted in second order, because as we know, the plant will not always show a linear graph. It is going to show peak in the in the number of leaves, or in the all other parameters that I have studied. But initially it is going to reduce. Initially, at the end, it is going to reduce as we move ahead with the experiments. So this curves basically determine micronutrient dose response patterns. It will involve deficiency, it will involve physiological optimum, and it will also talk about toxicity phases. So this graph is about number of leaves and concentration. The next graph is about plant height and concentration, where we can see the same nature of graph as we see it into number of leaves and concentration. For first 3 concentrations, that is uh, 50 mg, 25 mg per liter, 50 mg per liter, 100 mg per liter, the production has increased, but further it has reduced. The next graph is about stem girth and concentration. This graph shows that around 100 mg per liter, the Fe2O3 has some way supported the increase in stem girth, and further for higher concentrations, the stem girth has reduced. As you can say, for set 4 and set 5, it has reduced, and the red mark, it shows the control, which is 0 mg per liter again. Next is number of cloves versus concentration, and again, as we can see, for set 3, we have got a, a good increase in the number of cloves per per bulb of the plant, let's say. But further at group 4, for set 4 and set 5, as we can see, the number of cloves has decreased. So this represents Fe induced physiological stress and nutrient imbalance. This pattern reflects a typical micronutrient dose response relationship with deficiency and toxicity phases, as I've discussed earlier. Next, it is about fresh bulb weight and dry bulb weight. We can see the nature as curve, where we can actually say where we can analyze this as the 120 and 200 mg per liter mg per liter concentration has improved the bulb biomass. It is reflecting improved energy and metabolism and partitioning. But further, as we increase the concentration beyond 400 and 500 mg per liter, the yield has seen reduced. Even if the particle size is smaller than 100 nanometer, but even then, because of higher concentrations, we can see that fresh bulb weight as well as dry bulb weight of the garlic has reduced. So if I put the data, the final conclusion or the final result that we are expecting from this particular presentation is talking about the garlic crop productivity in terms of yield. So if we see that if we can see here in the chart, it is that the mean fresh weight has been calculated for all the concentrations for all the particle sizes, and we have compared the yield, which is into percentage, as to what yield I have received for control and what yield I have received for that particular concentration. And then you can see that in every concentration we have received more yield than the control. For 25 mg per liter it is 53 to 53.23%. For 50 mg per liter, it is 75.65%. For 100 mg per liter, it is 97.92%. For 200, it is 97.91%. And for 500 mg per liter, it is 51.91%. As you can see, we have received good yield, which is almost double for 100 and 200 mg per liter. If I decide to plot a graph between yield enhancement in percentage and Fe concentrations that I have used for all the plants that I have performed experiment on, then we'll get to see that 100 and 200 mg per liter concentrations have given almost twice the yield as compared to control. So in the conclusions, I must say that the yield has increased sharply for 100 mg per liter iron concentration, which is 98%. At 500 mg per liter for every set that I have performed experiment on, I can see that the yield has declined, which can be due to micronutrient toxicity or due to growth inhibition due to higher Fe levels being accumulated into the plant. The response is bell shaped, which we can also call as it is saturation type curve. Biologically, it is consistent with the literature review that we have done. It shows that iron is acting as an essential micronutrient, which actually is a micronutrient for plant. It is giving an enhancement plateau once the metabolic demand is met, and excess Fe can be seen interfering with the physiological processes of the plant. So this is all about my presentation in the conference. I want to thank the organizers of the conference for giving me this opportunity to present my doctoral research into this conference. Thank you. Thank you one and all.